Morning. My name is Aliz R, and I'm a bioinformatics researcher at UCLA. Today I'll be talking to you about a tool and methodology I've developed that allows researchers to calculate the statistical significance of their next-gen sequencing findings in studies involving Mendelian as well as complex disorders. First, I'll be going through the simplest case of how you would calculate a p-value if you've sequenced individuals causal for a Mendelian disorder and you want to interpret your rare variant findings. Next, I'll move on to some more complex cases and give a brief demo of the software tool. And finally, I'll go through some additional applications such as evaluating rare variants in protein domains and how you would use this tool for studies of complex disease such as autism. So to jump in, let me give you an example. Let's say you have a severe rare Mendelian disorder and you sequence 10 individuals with this Mendelian disorder, and you find that 4 out of the 10 have a rare loss of function or missense variant in the same gene. They're different variants, but they're within the same gene. You might think, wow, I might have found my causal gene. But what if I told you that 40% of the general population have rare loss of function or missense variants in this gene? Then your observations are exactly what we would expect by chance. But if I could tell you that only 1 in 1,000 individuals have rare variants in this gene, then your findings are a lot more interesting. And what's more important, knowing this population frequency allows you to calculate a p-value for your findings. So the answer we want to ask for every single gene is, how frequently do individuals carry rare protein-damaging variants anywhere in a gene? The model is that of a simple binomial model. Imagine that you have a sack of balls, and most balls are red. They indicate individuals in the general population, and a ball is white if the individual has a rare variant in a given gene. Now, what you want to know is, if you pull out, for our example, 10 balls at random, what is the probability of pulling out at least 4 white balls, given the frequency of white balls in the sack? Once you do this, you get your p-value. So what I did was, to get this frequency for every gene, I took the 1,000 genomes data. The phase 3 dataset contains individual level data for 2,504 individuals. I filtered variants by various filtering thresholds that researchers might use, and I collapsed variants by gene to get the count for every single gene. Now, in order to make this applicable to your research, you have to use the same variant filtering criteria in getting to these numbers that you would use in your study. So I repeated this exercise for various minor allele frequency thresholds. I repeated this for different models for disease inheritance. For example, if you're looking at an autosomal dominant disease, you will want to know the number of individuals with heterozygous or homozygous variants, both. But if you're looking at an autosomal recessive disease, you want to know how many individuals in the general population have homozygous variants only, or are potential compound hets, which would mean they have 2 heterozygous variants in the same gene. The 1,000 Genomes Project also has data from different populations. So I also took subsets of the data and calculated frequencies for each subpopulation in case a researcher would want to know the frequency of individuals only in Europeans or Africans, for example. Finally, I also did this variant collapsing for all known protein domains. Now, this ended up, if you take all these combinations of variant filtering criteria, a pretty large data set containing 840 files, and we released this data set under the name SORVA, for significance of rare variants. And most importantly, these values allow you to calculate the statistical significance of seeing rare variants in the constellation of families that you've sequenced. Obviously, browsing manually through these 840 files would be pretty cumbersome. So I've created a web tool, which is found at this first web URL, where you can simply enter your variant filtering criteria in a web form, and it'll give you the population level frequency for the individuals, as well as allow you to calculate your p-value. And I'll give you a demo in a few slides. There's also a standalone tool for submitting bulk queries or more customized queries, and that's available on GitHub. And finally, this manuscript is on bioRxiv, currently in its second round of peer review. So that should be out soon, and I invite you to check that out as well. So before I get into an example and demo, I also wanted to mention another method that allows you to calculate the p-value for your next-gen sequencing findings. Recently, a method has been developed called the transmission and de novo association test, which has been used in several recent studies on autism, for example. The downside of this method, though, is that it only works with trio and case control data. So I mentioned the simple model where you sequence unrelated, unaffected individuals, but one thing about this model is it even works, let's say, if you have a discordant SIB pair and you want to treat, then you treat that family unit as a statistical test. And if you, and you can even use it for pedigrees such as this. So this will be, I'll go through this as an example of how you would use the SORVA dataset to calculate your p-value. In this study of image syndrome, researchers did next-gen sequencing on 2 distantly related affected individuals and filtered down for the rare variants shared between them. And they also sequenced 4 unrelated, unaffected individuals. And they found that all 6 individuals that they sequenced had rare missense variants in one gene, CDKN1C, the gene known to cause Beckwith-Wiedemann syndrome. But all of these variants were missense variants in a very specific PCNA binding domain, and those are the black arrows seen on the top. Now, if you would do this analysis today, you might go to EXAC and see, take a look at the gene's PLI score and see that this region tends to be generally depleted of missense variation in the EXAC data set, and that's shown in the image on the bottom. But that still doesn't allow you to calculate a p-value for your findings. So I'll do a quick demo of the web tool, and let me switch screens over here. Oops. Okay, so if you go to the SORVA website and you click on Run a SORVA query, you would enter your variant filtering thresholds. So we are looking, considering all loss of function or missense variants. Our minor allele frequency threshold was less than 1%. We want to consider heterozygous and homozygous variants. And the gene of interest is CDKN1C. When we press submit, it'll tell us that 11 out of the 2,500 individuals have a rare loss of function or missense variant in this gene. That's equivalent to 0.4% of the population. And if you scroll down, you can see the number of individuals who have rare variants in specific genes as well. Now, if you continue to step 4 and calculate the significance of my variant, you can enter how many individuals you sequenced and how many you found, how many of the individuals carried rare variants in this gene. So we sequenced 4 singletons, and all 4 of them had rare variants in this gene. And let's say we had sequenced first cousins. So we sequenced one family in which we sequenced first cousins, and that family did have rare shared rare variants in the gene. And if you had and also if you sequenced any unrelated, unaffected individuals, you can count those as well if you use them for filtering out variants. What you want, the place where you enter the family depends on the coefficient of relation. So if you've had a SIB pair, the coefficient of relationship is 1/2. They share about approximately 1/2 of their genome. For first cousins, that's 1/8. And when you click submit, tells you that your p-value, after correcting for the number of genes we targeted, and we're putting 24,000 to be conservative, our p-value is approximately 4.9 times 10 to the -9 after correcting for the number of genes, which reaches genome-wide significant. So let me continue the slideshow. Okay. I also wanted to demo the Standalone tool. The Standalone tool is useful for running bulk queries. Let's say you have let's say you don't sequence as many individuals, or it's not as obvious or you have looser variant filtering thresholds and you have a pretty large candidate gene list. You can run queries on each of the genes. And if you have the relationship coefficients that weren't on the web tool, for example, if you have consanguinity in a family or larger pedigrees, you can have different relationship coefficients. You can enter the custom coefficients there. So the first thing you would do if you want to use the Standalone tool is, oh, yikes, sorry. Okay, you can download the Standalone tool on GitHub. It will allow you to, oh, sorry. You can download the entire data set with data on all 20,000 genes for all the 840 combinations of variant filtering thresholds. So it's a pretty large download, but once you do that, you can run one of 2 Python scripts, one to query the data files and get that population count, and the second to calculate the p-value. So in our example, let me do the same example that we did on the web tool. We were looking at non-synonymous or loss of function variants with a minor allele frequency of less than 1%. And the instructions are all in the GitHub README. And this will query the database and give that population frequency that 4.4% of the individuals have rare variants that meet those filtering criteria. And next, if you want to calculate a p-value, I wanted to mention that actually in the original research paper, the researchers were looking not at first cousins, but actually third cousins. So the relationship coefficient is actually 0.0078 instead of 1/8. And that's not, and you can't enter that on the web query. So you can use the Standalone tool to run this query. So you enter the population frequency from the first tool, how many individuals you sequenced that were singletons, how many did you sequence that had the variant, then for the custom relationship coefficient family, how many were there, how many had the variant, and then what was the relationship coefficient. And when you run that query, you get a value of 1.3 times 10 to the minus 14th. Now note that here I am not correcting for the number of genes that were targeted. So to do a simple Bonferroni correction, you would multiply this value by 24,000 or 20,000. And the real p-value will be closer to the order of 10 to the minus 10 or 11. So that's how you would use the Standalone tool, and you can see how easy it would be to generate bulk queries that allow you to run through all a list of different genes by just running the first command over and over, and then taking those frequency counts and plugging those into the second command. So let's go back to the presentation. And I won't be doing any more jumping around windows after this. Okay, so in conclusion, by running this tool, you can calculate, you can discover that 11 out of the 2,500 individuals have rare missense or loss of function variants in our gene of interest. And most importantly, it allows us to calculate a p-value for our findings. And yeah, so the easy question to answer is, what is the frequency of a given variant? For that, you can browse the EXACT data set or the NOMAD data set and answer that very easily. But what's harder to answer is, how frequently do individuals carry rare protein damaging variants anywhere in a gene? And that's what this SORVA tool allows you to answer. So in summary, knowing how frequently individuals carry rare protein-damaging variants anywhere in a given gene allows us to, 1, prioritize gene lists, and 2, calculate the significance of our findings. Now, I wanted to jump into some additional insights and applications. This was it for the demo, but if you're interested, stick around. So this mountain of data we can browse this mountain of data and reveal a couple of interesting insights about population structure and variation in general. So for example, one thing we noticed is that if you change your variant filtering thresholds by minor allele frequency, the number of genes that contain rare variation doesn't change by that much. So it's not critical to choose the right minor allele frequency threshold to get an accurate p-value, but what is very critical is to determine your protein consequence threshold. So if you're filtering for loss of function variants versus loss of function or missense variants. For obvious reasons, loss of function variants are a lot more rare. So if you're filtering for only loss of function variants, you're really enriching for disease variants. unfortunately, with this size of data set, many genes will contain actually 0 loss of function variants. So what you can see in these charts is that for the 20,000 genes, most genes contain data, and individuals will carry variation in all populations, or 4 out of 5 populations. But a small fraction of genes don't even contain missense variation. But if you filter for loss of function variants only, you find that actually about over half of genes contain 0 loss of function variants. So that becomes a problem when you try to calculate the p-value. You have a 0 numerator problem. So when you actually calculate p-value for these genes, you want to use an f that is very small. And you can even get a better estimate by looking at the exact data set and just using some of the numbers from the exact data set to estimate this f. By the way, at this point, you might be wondering, why didn't I use the exact data set for this study? Well, it's really important to have individual level data, and exact doesn't release their data per individual for each of the 60,000 individuals, because if you have more than one variant in the same gene in an individual, you don't want to count them twice. So for some of the more frequently mutated genes, this would not result in good numbers. So additional things you can look at, if we look at the number of individuals who carry rare variants in a gene in different subpopulations, we find that Africans tend to have higher numbers, and that's expected since they have 3 times, Africans tend to have 3 times as many rare variants as individuals in other populations, such as Europeans and East Asians. There are population differences between these counts. However, for most applications, I would recommend you just use the entire data set, because if a gene contains rare variation in Africans, it still probably doesn't cause rare Mendelian disease. Next, just as a sanity test, I looked at whether genes known to cause Mendelian disorders tend to have fewer individuals with rare variants in the gene. And we find that that is the case. Here I'm plotting the genes along the y-axis is the number of individuals with rare variants in a gene. If you filtered for heterozygous variants with a minor allele frequency of less than 0.5% that are loss of function. And the colored shapes indicate the centroids of the genes in each category, OMIM disease category. So for example, down at the bottom, the yellow diamond is the centroid for all genes known to cause X-linked disease in OMIM. And those are generally very depleted of rare variation. You see that the diamond falls between 0 and 1. So those genes tend to be depleted of variation in general. Another thing we can look at is how many individuals have rare variants in a given protein domain. And this is very useful in the case of our example, where we found that all protein domains, all of the variants in our gene of interest fall into the same protein domain. And some additional things you can look at, you might wonder, well, are any So what protein domains in genes tend to be depleted of variation compared to the rest of the gene? So we found that in our data set, the genes above the red dashed line had protein domains that were depleted of variation compared to the rest of the gene. So those might pinpoint functional protein domains that if you find a rare variant in, then that might indicate disease. It might indicate that those variants are more likely to cause disease. And another thing you might want to look at is a certain type of protein domain generally depleted of variation. So many genes will contain, let's say, family AG protein coupled receptors, and this data set is available in a supplemental file. I'll run through a quick example here how you would use this. Let's say, let's go through this second line as an example. You want to know, are family AG protein coupled receptors depleted of variation in general, in the general population? So if you want to ask that question, in this data set, you can see that, first of all, there are a total of 874 instances of this protein domain in 660 different genes. So, and these 660 genes have rare variation in approximately 4 out of the 2,500 individuals on average. And yet, the variant is never in the protein domain. The variant never falls into this family AG protein coupled receptor. And that's this fifth column showing 0. And this isn't a small protein domain either. The protein domain is almost 1,000 base pairs in length on average in these 660 different genes. And it's well covered. In the 1,000 genomes, in one individual in the 1,000 genomes data set, it was covered at over 100x, and more than 80% of the bases were covered at over 10x. So this is highly indicative that this type of protein domain is depleted of variation in the general population. So if you're ever sequencing a disease, sequencing individuals with a rare disease, and you find that the variants fall into this protein domain, that would make your findings a lot more interesting. So whatever protein domain your variants fall into, you can query this table and find that out, and manually you could even calculate the p-value. Now, I did want to compare SORVA to some other existing methods. Now, other methods to calculate the p-value of your findings aren't available other than the transmission and de novo association test, or TADA, but there are other methods that allow you to rank genes. So I compared SORVA to how, so the order that SORVA would rank genes at a given combination of variant filtering thresholds. I compared it to how exact PLI scores would have ranked the genes, as well as eVOTOL, FLAGS, and RVIS. SORVA is in these ROC curves at the bottom. SORVA is the red line, and I'm showing, I'm in the first plot, I'm showing variants filtered for an autosomal dominant model. In B is an autosomal recessive model, and in C is an X-linked inheritance model. And SORVA contains, SORVA tends to, and what I'm showing in all three of the ROC curves is how many genes known to cause disease are ranked up to a certain point by each of these methods. And I showed that SORVA does perform comparatively to the other methods, depending on the inheritance model. For X-linked diseases, it does second best only after the exact PLI score. And the ordering of the genes is actually most similar between the exact PLI score and the SORVA scores. Now, let me jump into a more complex case. What if you're looking at not rare Mendelian disease, but complex disease such as autism? Now, there are possibly hundreds of genes causal for autism, and they generally fall into a couple different pathways, and this is just starting to be revealed in recent years with all the next-gen sequencing going on. But what we did was we wanted to compare how SORVA would have done in calculating the significance of some of these findings. So we compared the gene list that found by one recent study in which 44 candidate autism genes underwent targeted sequencing in over 2,000 individuals. And in the initial study, they found that 6 genes were statistically significant, and these researchers did simulation studies and simulated the mutation rate in the genome to get to these p-values. So the 6 gene names that this study found significant are in the marked in bold in the first column. And if you enter the number of individuals with that they found rare loss of function variants in into SORVA, we can calculate a p-value as well for their findings, and the genes marked the genes we found significant using the SORVA method in almost all cases match up to what the researchers found were significant. We find that 5 out of their 6 are significant using SORVA. One that was borderline significant in their study didn't meet the threshold for SORVA, although we did find that one additional gene, the second gene, ADNP, was significant using SORVA because we're incorporating the information that this gene is extremely intolerant of variation in the general population, and that makes this gene significant. But generally, it's pretty comparable. The results are comparable to what you would have gotten if you did simulations. And that just shows that you can use SORVA to calculate p-value similar and get similar results if you did simulations, but just in a much easier way by simply using the web tool or the standalone tool, because we had pre-computed the values that allow you to get this p-value very easily. One more thing we looked at, and I'm just going through the different sections of the manuscript. One other thing we looked at.
Aliz, it's Dennis. Just it would be great if we could close like in 2 minutes.
Oh, okay. Okay, so let me jump ahead and really quickly, I did want to mention a really exciting application. You can actually use the SORVA score in predictive genomics, where if you find a rare variant in a known disease gene, you can actually calculate the probability that that person would develop the rare disease. Let's say if you're doing prenatal testing. And I won't go through the actual numbers in this example, but what you would find is that let's say you find rare variants in POM GNT1 that cause congenital muscular dystrophy, and you plug in the numbers, and this the denominator has the SORVA score, and you can find that if you have compound Het in this variant, you have a 1% chance of developing congenital muscular dystrophy. So I just wanted to highlight that due to the extremely low numbers for the prevalence of the disease and how rarely these genes contain rare variation, even if your compound Het for this known disease gene, you have a very low chance of developing congenital muscular dystrophy. Obviously, this would be for prenatal testing. If you were born, you'd already know. So in summary, the SORVA dataset is widely applicable to researchers to calculate p-values for their next-gen sequencing findings. The scores are directly interpretable, and it's a lot simpler than running simulations. So I invite you all to try it in your next study if you are doing rare variant interpretation. Okay, and thank you. I would just like to acknowledge, oh, sorry, not all of that applies. So thank you, and I will be happy to take some questions.